Okay, here we go. Cell division. So the previous video was about cell division in general, but it was really more about kind of the structures of chromosomes and things like that. I think I would title it a little differently because really this is the video on the actual dividing, which we might call the cell cycle. And again, I'm basically following Clark, Choi, and Douglas Biology 2E. Open sourceware out of Rice University. Okay, the cell cycle. So if you've had high school biology, or maybe you're in high school biology, you know the basic stages of the division of a cell: mitosis, gesundheit, mitosis. But anyway, the kind of in between time is called interphase, and you can see that interphase, most of a cell's life is in interphase. Most of the cell's life is getting, well, I don't know, kind of in between. Mitosis is a fairly small segment, as you can see, the mitotic phase, where you get the formation of two daughter cells from one parent cell. That most of that of a cell cycle is basically growth. DNA is replicated in the S phase. You might remember from the previous video that the DNA doubles itself, it replicates itself into two homologous chromosomes, two chromosomes that look exactly alike. I understand that now better when we get to this video. That happens in the S cycle of interphase, but the cell is pretty much intact and everything's in the nucleus, and then the nucleus starts to break down at the beginning of mitosis. Not to be confused with halitosis, which is bad breath. I went to Starbucks. No, I didn't. I went to Barnes & Noble that has a Starbucks in it today. And as I was checking out, they said, would you like some Altoids for $1? I'm not sure what that means. It might be because I have mitosis or halitosis, one of the two. Okay, so whoop, I split in half right there. So mitosis is a very small segment of the cell's life. Cytokinesis, think of cytoplasm, gooey. The cytokinesis means to move, kinetic energy, So cytokinesis is the movement of the cytoplasm. This is where it actually splits. This is where we've got two baby daughter cells. Cytokinesis, the movement of the of the cytoskeleton and everything. That's where it splits. Interesting that it has it pictured as roughly taking the same amount of time. I would have thought of cytokinesis as much smaller. Maybe it's not to scale. Doesn't really matter, does it? So this is interphase, the in-between time, with DNA replicating in the S phase. I know that's something that you're going to need to know. And mitosis is the separation of basically the stretching into almost two, and then cytokinesis is where we've got two babies. Okay, so that's an overview of the process. Interphase, mitosis, cytokinesis. Interphase, in between the phases. Mitosis, the splitting, and cytokinesis, the movement into two. Okay, there you have it. By the way, it doesn't always end in cytokinesis. There are, have you ever had an egg with two yolks? You open it up and there are two yolks in this egg. Well, you do have sometimes a kind of splitting of all the insides without the actual pulling apart of the cells. That can happen, but this is the normal process right here. Interphase, mitosis, cytokinesis. Interphase, which you can't see because of my face. So if you remember this diagram, there were three phases of interphase: G1, S, and G2. I'm not sure where those come from because I'm not a biologist. I'm just making videos. And so G1 phase, nada seems to be happening. So if you were looking under a microscope, it looks like nothing's happening, but they are gathering stuff, okay? You got some people gathering sticks out there, some protein. I'm gathering proteins. Oh, look, there's some lovely proteins over here, Dennis. But anyway, they're gathering stuff and they're getting their energy up. They're doing their calisthenics, getting ready for the race, jumping jacks. Okay. So, but nothing seems to really be happening in the G1 phase. There is on the biochemical level, on the small level, it's getting ready, but nothing seems to be happening in G1. S, again, important to know, this is when the DNA replicates itself and we get the chromosomes. So in the early, in the G1 phase, the chromatin fibers, the DNA's kind of hanging out. It's wrapped up. It's just kind of hanging out at the bar. What did you do this weekend? So that's the G1 phase in the chromatin fiber kind of thing. But by the end of the S phase, we've paired up into these, remember, chromosomes, homologous chromosomes, 2 chromosomes that are exactly alike, and they're tied in a nice little Christmas bow. They're at the centromere, isn't it? Centromere, isn't that what it's called? Centromere, oh boy, I hope that's right. Let's look. I can go right back because I've got a video. There, centromere, yes. They're tied up at the centromere to chromatid. We've gone from chromatin fibers to chromatids, 2 identical homologous chromosomes. These are they're ready, tied up in a little centromere bow, getting ready. Okay, so my brain has not failed me yet. It will. I got to get it all out before it all seeps out of my holy brain. Okay, so that's the S phase. So G2, so the centrosome splits and gets ready in this. So we do have the centrosome. So what are centrosomes? Centrosomes are these 2 tubes that are kind of hanging out outside the nucleus. They just kind of a centrosome. What is it doing here? What are you doing here? Who let you in here? It's just hanging out. In the rest phase, in the G1 phase, it's just kind of hanging out. But in the S phase, as the DNA replicates, this centrosome splits into 2 centrosomes. What this centrosome is going to do is it's going to, the centrosome is going to pull the stuff apart when we get to anaphase. Anaphase, tug of war. Anna's in tug of war. She likes to play tug of war. They're 2 Annas. They're identical twins. In anaphase, we're going to pull the stuff. We're going to pull it apart in anaphase. So the centrosome seems to play a key role in that pulling apart. And so in the S phase of interphase, the centrosome splits and it's getting ready. We get little mitotic spindles. A spindle. These are the webs, the spindles that are going to. Remember in what was it, Maleficent? All those spindles. Anyway, maybe that's wrong. But anyway, the spindles are going to be part of the. Think of the spindles as the ropes that are pulling it apart in anaphase. But we're getting ready in the S phase. We're duplicating the DNA. And we're splitting the centrosome, and we're getting ready. Okay, don't confuse the centrosome, which is the puller, from the centromere, which is that little bow that ties. It's a mere bow. It's just a mere bow that ties together the 2 chromatids. Then the G2 phase of interphase, it's getting charged up. It's getting ready to rumble. It's the organelles are doubling. 1 mitochondrion becomes 2 mitochondrion. 1 endoplasmic reticulum becomes 2. It's getting ready. Waiting orders, ma'am. G2 phase. Okay, so again, this is all interphase. G1, gathering energy, but it doesn't look like anything's happening. S phase, replicate the DNA, replicate the centromere, starting to get out the spindles, and then we're energizing, getting energy, getting ready, stretching, getting ready, and that's all interphase, G1, S, G2. Okay, interphase. By the way, this is a place I'm going to put in G0. I know this is a study question somewhere. G0 is not go. It is not go, okay? So it's a no-go phase. I put empty set. No, okay. G0 basically means we are not, halt, wait, we are not replicating. So if something is in a G0 state, it is not going to replicate. It's waiting. So we don't have enough energy. We need some more energy. Okay, we're going to just put this on hold, okay? We're not splitting today. Don't split. And so if something is in a G0 phase, it's not splitting. It is a no-go if it's GO. So GO is a holding pattern. Do not replicate. Do not replicate. So, okay, it's a no-go if it's GO. By the way, there's some cells that are permanently like this. Once your heart is mature those cells don't replicate. Once your nerves are mature, they don't replicate, which I wish they did. I mean, I wish I could sure use a few more brain cells, frankly. They're quiescent. In the no-go phase, they're quiescent, which means they're quiet. They don't replicate. They're done. So this is something that I wanted to throw in here, and this seemed as good a place as any. Okay, so we've talked about interphase. Now let's talk about mitosis. So mitosis is, again, this is the actual kind of splitting part. Another name for it is karyokinesis, which I guess they do karaoke music while they're doing this, while they're moving. Kinesis, they do karaoke in mitosis. Spin your partner around, you know, they're getting ready to pull apart. And so that's karyokinesis is another word for mitosis. Now, the stages of mitosis are 5. I think they've slipped one in here since I was in high school. I don't remember one of them. One of them seems new. So I know prophase, and I know the 4 last ones, but the second one, prometaphase, I don't remember that. When did they come up with that? Cells don't divide the way they used to when I was a boy. Anyway, but prophase, prometaphase. Let's go ahead and get it all out here. Prophase, prometaphase, metaphase, anaphase, and telephase. Okay, so this makes a little sense to me. Pro means before in Greek. I'm trying to think of a good derivative for this. A proposition is something that is put before you. That doesn't really help. Pro football doesn't help, right? Because that's the advanced one. So that doesn't work. But pro can mean before. Can you think of one? There are English words, pro something, that mean before something. It's a Greek word, pro, means before. Anyway, so prophase is before. It's the early, and then prometaphase is before metaphase. So that all makes sense. Metaphase is where they're lining up in the middle. They're getting ready to do the hoedown. Sprint your partner, you know, the kind of do-si-do, you know, they're getting lined up there in the middle to get ready for it. Then anaphase, as I said, is the pull. And then telephase is the end. Tele means goal or end, by the way. If you knew Greek, if you only knew Greek, this would be easy. So the telos of something is its goal. And so telephase is the end phase. So, okay, so how does this work? So prophase is everybody to your stations. So getting ready. We're getting ready to rumble. That's the prophase. Prometaphase is where hook up the spindles. Okay, so they haven't really started. These are the prophases. They haven't really started. So everybody's kind of, okay, let's get in the room. Come on, stop talking. Everybody get ready, prophase. Okay, come on. Over here. Hey, stop talking. Get over here. Okay, so prophase, everybody's getting ready. Prometaphase is, okay, we got to hook up the spindles. So they're hooking them up the spindles to the chromosomes, hooking up the spindles. And then the metaphase is line up. So everybody lines up in the middle. Fill in. You know, they all get in the called the metaphase plate in the middle. The chromosomes all line up. So for human beings, 23 partners are lined up to get ready to pull apart. 23, is that right? I guess it should be 46, shouldn't it? Because we're not talking about gametes here. We're talking about actual cells. Yeah, so 46 chromosomes lined up. Okay, and you can see them, by the way, at this point with a microscope, not with an advanced microscope, but with a good microscope. You can see it. So they line up in the metaphase, and then again, anaphase, tug of war, pull, with Anna playing tug of war. Okay, pull, and then telephase. Okay, reassemble, get in your corners. So that's kind of the way it happens. Now, there's some lovely little diagrams in there. Prophase, again, getting ready to rumble to your stations. So what happens in prophase? We're getting ready for mitosis, the first part of mitosis. The chromosomes kind of contract, they condense, they become visible. Spindle fibers begin to emerge, these little spider fibers. The nuclear envelope is breaking down. So we want to release the chromosomes. And so the nuclear envelope begins to, see the dotted means it's breaking. We're breaking up down here. Nucleolus disappears. You know, so no more nucleolus. So that's all prophase, okay? Getting ready. Prometaphase, again, we're bringing out, are we hooking up? Yeah. We're grabbing the spindle. We're hooking up the chromosomes to the spindles in prometaphase. Kinetochores are, so here's what's happening. We're attaching, in prometaphase, we're attaching the mitotic spindles. These microtubules are hooking up, like getting ready for the tug of war. And this is the centromere region here in the center. These are sister chromatids. And then the kinetochore, the movement core, that's kind of a protein, I think, it's like a glue or a hook. I mean, think of it as a hook. It's where we're hooking up the spindle, the kinetochore. We're hooking it up right there in the middle. Bipolar microtubules are the microtubules over here that are going to help kind of pull the cell apart. But polar means at the poles. So the polar microtubules are closer to the poles of this thing. Kinetochores are again the kind of the glue proteins that glue it on. Grab a spindle, okay? Centrosomes are moving toward to the side so that they can get ready to pull apart. Okay, so metaphase. Line up, everybody line up in here. So this is the metaphase plate. Everybody's lined up in metaphase. All the spindles are attached. So the mitotic spindle is fully developed. Centrosomes have gone to their opposite poles. These are the centrosomes, I think. The metaphase plate, everybody lined up. Each sister chromatid is attached with the kinetochore, and everything's ready to go. So prophase. Okay, everybody, quiet down. Everybody, get ready. Everybody, okay, come on. Prometaphase again. Attach your spindles. Okay, prometaphase, attaching the spindles. Metaphase. Okay, everybody, line up. Fall in. Okay, everybody's ready to go. And then anaphase. Pull, 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 pull. So ana is pulling. The cohesion proteins break down. So remember, the cohesion was holding the chromatids together. It breaks down. And so now these sister chromatids, which are homologous, they're the same, are pulled toward opposite poles. Okay, and these spindles elongate the non-kinetochore spindles that I called, what did I call them? Polar microtubules, they begin to lengthen the cell here in the middle. So anaphase, pull, 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 pull. Telephase is the end. Okay, we now begin to form two nucleuses, like two yolks in an egg that had two yolks. Telephase, we reassemble on each side. Things begin to we now have two, the materials for two cells, one on each side of this elongated thing. The mitotic spindle now is no longer needed. We got them. Okay, unattach. And so we are ready for cytokinesis, which is where we now we close in the cytoplasm. The little difference, we split the goo in cytokinesis. It goes to a different end here. So plants have a wall that forms between them, whereas the animal cells more just kind of get a cleavage, and then bloop, it pulls apart. Whereas the cells stick a little bit closer together with a what's called a cell plate in the plant in between it. Okay, well, we got through it, and I didn't, in my brain, I didn't drop anything from my brain. I don't think, oh, there is something on the floor. I'll get it after this video is over. This has been the cell cycle with a little bit of interphase and mitosis and cytokinesis, the splitting of the cells.